I am a PhD student here at KAVI, a PhD in Immunology. So KAVI generally is an institute that does a lot of clinical trials for HIV vaccines, trials and other infectious diseases. And we also do a lot of basic research work, all to inform on how to come up with effective vaccines for different infectious diseases. And my work mostly tries to understand the female genital tract, because the female genital tract plays an important role in women's health. For example, in HIV transmission, studies have shown that women are at a higher risk of HIV acquisition as opposed to men. And this has been linked to the female genital tract structure, first of all. And that's why I'm more interested in understanding the female genital tract, because it plays an important role in women's health. And also in things like pregnancy outcomes, the female genital tract plays a very critical role. And that's why I decided to do a project focusing on women's health, particularly focusing on the female genital tract. So my study tries to understand how alterations or how different microbial communities in different women might affect the integrity of the vaginal secretions, which form the first line of defense upon exposure to any pathogens. And in terms of next generation sequencing, so like now I use the MiSeq i100 for the sequencing. So that one helps me to be able to at least know the different communities that I have in these samples. And then the beauty with the MiSeq i100 instrument, the cartridges come at room temperature. So I don't have to do the thawing bit, which I would have done with the old MiSeq. So I just have the cartridges in my packaging and then remove them. The most important thing to note is that you only have to open that pack within 4 hours. So you have to make sure you don't open it earlier. So once I open the cartridge, I just need to load in my sample that I've brought to 60 picomolar, and then I put it into the machine. So with the MiSeq i100, the process has been shortened and it's like very easy, straightforward to do. And also the data acquisition, it takes shorter time, maximum of 15 hours, unlike the old MiSeq where it will take like 48 hours. Adopting the MiSeq i100 in this lab will definitely improve the research work here. First of all, for example, for the old MiSeq, we could only do 1 run per week, because given the time it takes to give out data. So we could only plan 1 assay per week. But given the new MiSeq i100 that only takes 15 hours, we can do even up to 3 assays in a week. And then also the quality of the reads you get with the MiSeq i100 are more. And this translates to better data and therefore better clinical results that you can give out to the patients out there. I think we are heading into the right direction, because first of all, we are going to be getting quality data which translates to like quality implementation into the society. And also I think we are going to have like more knowledge comparable like to the Western countries that have always been known to be doing more of this next generation sequencing. Like previously, you'll find most of the samples will be sent outside for sequencing. But now with these new technologies coming in, like the MiSeq i100 from Illumina, which has a very straightforward process, like anybody, so long as you have the protocol, you can follow it. So I see most of like us researchers in Kenya being able to do quality next generation sequencing work and producing quality data that is going to improve generally our health. I'll just encourage our researchers not to be afraid to do such work, because it's possible, and we have the technology, we have the instruments, and we have the support from the companies. You know, in case you are stuck anywhere, it's just a phone call away and they will come and give you the necessary support that you need.